By nature, fishing is a non-random process. Typically, gear is designed with specific species in mind, and therefore the removal of unwanted species in preference to others can occur. Similarly, the location in which fishing occurs is non-random. Spatial distributions of fish stocks are known due to development in water column wavelength imagery. This spatially exploits stocks which are easily harvestable and where biomass is greatest. There are many evolutionary effects of fishing, but these mainly manifest in two ways. Firstly, due to design, nets are size selective. The mesh sizes are made functionally in order to catch differently sized fish. Also, due to similarities in morphology, not all fish of a certain size are necessarily the target species. This creates bycatch and an unnatural selection pressure on fish size. Secondly, changes in behaviour can be observed. Many fishing practices lend themselves to catching and targeting more inquisitive and bold fish, resulting in a selection pressure for more timid and shy fish. These artificial selection pressures imposed by fishing are opposite to those which are seen naturally. It has been seen that rates of fishing mortality are different to natural mortalities. In the case of fishing, mortality will increase with size, as larger fish are typically size selected for value, and also more frequently caught in nets, even as bycatch. It has been seen that current methods of fishing are having an influence on life history traits of exploited fish stocks. This is generally seen via changes in size and ages of fish at maturity, differences in reproduction and fecundity, fluctuations in recruitment and survival, and finally a broader ecological effect on population structure and the surrounding ecosystem. The size and age of individuals at first maturity is an important indicator of the current state of life history traits in fish stocks. By assessing stocks' average size and weight at first maturity, the patterns can be tracked to identify the changes in the life history traits. Variations between R and K selective individuals are evident when observing the size and weight at first maturity. R selective traits focus on a small size and young age at first maturity in order to maximize reproductive output. K-select species trend towards larger sizes and older ages at first maturity, maximizing reproductive efficiency. The age at first maturity is not the only trait needed to be shown by a species to be considered either R or K selective. However, evidence has shown that these simple parameters can help to suggest more complex parameters that are more indicative of those life history traits. When comparing the age at first maturity, any factors that influence the trait must be considered, such as the temperature of the environment. Lower temperatures have been shown to increase the age at first maturity for a wide range of fish and is therefore paramount to consider when investigating life history traits. Published review articles have discussed the shift in skate species where prior to industrial trawling, a roughly equal mixture of larger species with later ages of first maturity and smaller species with earlier stages of first maturity were seen. The effect of these trawls has significantly reduced the abundance of skate species with later ages and larger sizes at first maturity. Going further, some experts have concluded that having a lower age at first maturity in shark species was the strongest impactor on rebound potential. In many fish species, fecundity can be seen to increase with size and also age, such that mature adults add a high reproductive value as they have a higher probability of contributing to population growth. Many fish, such as those in the families groupers, Sirenidae, and rockfishes, Scorpinidae, show high fecundity, producing thousands of millions of eggs each spawning season and several batches of a couple of million of eggs each year respectively. However, high fecundity is not the only factor responsible for population maintenance during heavy exploitation, as the size and age at first maturity is key to whether or not a sufficient proportion of the recruits are able to reach reproductive age and size before being removed from the ecosystem by fishing vessels. As previously mentioned, if a species is forced to shift to more R-selective traits by the effects of overexploitation of fish stocks, the size at first maturity will reduce, which in turn is capable of reducing the fecundity of effective species. For example, the reproductive output of a size-selectively harvested marine limpet inside an MPA was found to be 80 times higher than exploited population. In hermaphroditic species, in which all individuals start as the same sex and then differentiate at a later stage, if adult mortality rates are unnaturally high due to size-selective harvesting, the age in which they differentiate will be reduced to compensate. This has been seen in longfin eel populations in New Zealand, where sex is typically environmentally determined, such that males will differentiate at a younger age and smaller sizes than females. With males falling below minimum catch size in the area, it is the females that are typically targeted in fisheries, leading to an extreme male bias, as differentiation to males is favoured in an evolutionary response to fishing pressures. Another important factor in the changes of life history traits of farmed fish is the recruitment rate. Recruitment in ecology is defined in the 1996 Annual Review of Ecology and Systematics as the addition of new individuals to populations or to successive life cycle stages within a population. Recruitment overfishing, where the fishing of aquatic populations causes the reduction of recruitment within those populations, has been called to be noted by fishing managers and conservationists. The conditions of large population and local recruitment are likely to generate adaptive variation at finer scales, although the data available so far suggests that the scope for sweepstake recruitment, which is where entire families and groups either survive or die, may be higher in larger populations as the ratio of genetically effective population size to the total adult population size is lower in large populations. Even assuming extensive sweepstake recruitment, low effective population size over total population size ratio may be puzzling, given that the genetically effective population size depends on lifetime and not annual reproductive success. It has been discovered that the rate of recruitment is related to the abundant spawners. The highest rate of recruitment has been observed to occur when the abundance of spawners is at its highest, and vice versa. Therefore, spawner abundance cannot be ignored in the management of fish populations, which in turn highlights the problem of recruitment overfishing. A case study looking at recruitment and stock recruit residuals within the Gulf of Alaska and the Bering Strait Aleutian Islands showed significant positive covariation within several groups of species and significant negative covariation between certain others. This, and other recent studies, demonstrate that the recruitment and survival of different fish stocks in the Northeast Pacific does not fluctuate independently, but instead shows significant positive or negative covariation. Evaluating the impacts of fisheries on marine communities is difficult due to there being very few or no control sites where fishing has not been undertaken. The awareness of the undesirable effects that fishing of commercial species has on the surrounding marine ecosystem has only recently been reached on all levels of the fishing community. Fisheries can transform a stable, mature and efficient ecosystem into one which is stressed due to changes in life history traits that have been previously discussed. Changes in the life history traits of fisheries can have ecological consequences leading to large changes in the community composition and functioning of the ecosystem. Some of the problems that overfishing can have on ecosystems include reductions in the species diversity, a decline in the aggregate production of exploitable resources, and an increased bycatch and increased anthropogenic habitat modification. Adjusting normal predator-prey interactions can influence marine food webs and trophic interactions by removing primary prey for the higher trophic levels or predators of lower trophic levels, having cascading effects on the food chain. Shifts in the recruitment, survival and reproduction can have significant implications for the future of fishery populations by causing a reduction in the population stock and its growth. Poor recruitment, survival and reproduction due to overfishing can lead to loss of native species, decreasing the overall biomass and yield of fish stocks. Generally, problems facing marine resources such as fisheries are often due to the failure of management. For example, fishery management and conservation are often too narrowly focused on a single species stock and its control strategy. Knowledge and increased understanding of life history parameters including fish stocks will provide a starting point for the management of fisheries.